Assalamu alaikum and a very warm good evening, everyone. I hope you have enjoyed all 5 days of 1 week AICTE-ISTE sponsored faculty induction program on multidisciplinary and translational drug development. I am Abu Sufyan, Assistant Professor at School of Pharmacy and Event Lead Coordinator on behalf of Anjuman-I-Islam Kalseka Technical Campus, New Panvel. Extend my warm welcome to all dignitaries, resource person, faculty members, participants from different prestigious colleges across the India to concluding day of this refresher program. To start with, as a tradition of Anjuman-I-Islam, I will recite and translate few verses from Holy Quran. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم ولتكن منكم أمة يدعون إلى الخير ويأمرون بالمعروف وينهون عن المنكر وأولئك هم المفلحون. And let there always be among you a body of men who should invite you to goodness and enjoin virtue and forbid evil, and it is they who shall prosper. Sadaqallahu Azim. These verses from Surah Al Imran give us formula to prosper in the life here and hereafter. Indeed, it is worth implementing in our life. Yesterday, we had very enlightening session on breakthrough in drug delivery technologies, application of 3D printing by Dr. Tahir from Nottingham University, Malaysia. In his session, he discussed about feasibility of use of 3D printing in formulation of different dosage form, bioprinting, and also about future change in dosage form manufacturing in line with this technology. Going ahead, today we have very relevant session on antibody for COVID-19, learning from product development. For this session, we have with us eminent resource person, Dr. Kafil Ahmed. It's my privilege to introduce him. Dr. Kafil is a strong research professional with PhD in biotechnology. He is experienced scientist with demonstrated history of working in biopharmaceutical industry. He is skilled molecular biologist having experience in donor-based Navy SCFE antibody library construction, antibody gene cloning, expression and screening, antibody engineering, and lead generation. He having experience in development of biosimilars, monoclonal antibodies, and other therapeutic proteins like insulin and its analog, erythropoietin, GCSF, and interferon. He worked with Glenmark Pharmaceutical in biological department as a senior research scientist. He has experience in discovery and development of antibodies for the treatment of various type of cancer and autoimmune diseases. He was part of team who have launched a recombinant insulin in the year 2004. Currently, he is working as a director of Resino Biotech. With this brief introduction, I extend my warm welcome to Dr. Kafil and I request him to start with his session. Dr. Kafil, it is over to you.
Hello, Assalamu alaikum.
Islam.
Good afternoon, everybody, and thank you, Dr. Sharik Saab, Dr. Abu Sufyan Saab, for giving me an opportunity to speak about, COVID antibodies, how it is, how we can develop it, and then what are the uses? How much beneficial it is for the, for the common man. Can you? I'm not able to see the slides.
You'll have to share screen again.
Yeah. Okay. Is it visible to everybody now?
Yes, visible.
Okay. So first we will, I will speak. I will just explain about general antibody development, and there is some slight difference between discovery of COVID-19 antibodies. So what type of different platforms are there? What is the process, and how discovery from where it is started? Discovery start. So as everybody know that discovery of any molecule, doesn't matter whether it is a chemical molecule or biological molecule, it is really tough to discover some molecule which are which will be useful for any type of disease treatment. So as you know that the discovery, so we will. I mean, right now I will speak about just the overview of antibody discovery. So this is just the structure of antibody. So what are antibodies? I will start with the basic of antibodies. And in between, if anybody have question, you can just interrupt me to make it more interactive instead of just I will just give a walk through the presentation. So in between also anybody can ask any type of question related to antibodies, general antibody, or maybe a therapeutic antibody, or COVID. Why there is a need of COVID antibody, something like that. So antibodies are protein made in the body in response to foreign particles such as bacteria and viruses. So it is produced by your immune system, B cells, if there is any infection. So there are two different, classes of antibody. I could say that polyclonal and monoclonal antibodies. So polyclonal antibodies are mainly used in, were used in, long, maybe 10, 15, 20 years back or 30 years back for diagnostics. So mostly the therapeutic antibodies are monoclonal antibodies, which binds to a which binds to a single epitope. So this is the target antigen, this is the antigen, and this is the structure of antibody which binds to a specific antigen. So it doesn't cross reactive to any other epitope or any other antigen. So this is in general the structure of antibody. What type of so there is a heavy chain and two heavy chain and two light chain, and this the weight of molecular weight of one heavy chain is approximately 50 kd kilodalton. Then this is 50 kilodalton, and then this part is a light chain. This blue part is a light chain. So the molecular weight of this is 25 kd, and this is 25 kd, and this region is called as the Fab and the Fc. So this part, basically the variable heavy and variable light chain, it basically binds to any epitope, and this part gives the stability, the Fc and other, to the protein molecule. So there are different types of immunoglobulins produced in our body. That is IgM, IgG, IgD, IgE, and IgA, distinguished by their constant region responsible for effector interaction. So antibodies are very specific. Total molecular weight is 150 kd. So what are the different platforms for developing a monoclonal antibodies? So there are 3-4 different platforms which are widely used to discover novel antibodies against any given antigens. Maybe it doesn't matter whether it is a cancer target or whether it is autoimmune target like ankylosing spondylitis, rheumatoid arthritis. So there are three, 4 different platforms. So one is the very old platform, that is a hybridoma technology. The second platform is antibody fast display platform. So I have worked on this platform for 10 years. So I don't know much. I do not have any practical experience on working on hybridoma or yeast display platform. My expertise is to work on, mainly I have worked on fast display technology. So I will speak only about the fast display technology and some little bit about the hybridoma technology. So this is the old technology. It's called as the hybridoma technology, where you need to just inject the antigen, where any target antigen for which you like to develop antibody, and then you have to, once you have done this thing, then you have to isolate the spleen and then fuse the cells with the myeloma cells and then select the cells, and then these cells are called as the hybrid ones. Once it is uh, you know uh, and then these hybrid cells has to be selected on hybrid media. It's a selective media, only the hybrid cells are selected, and then you can do a ELISA for the isolation of a specific clones, antibody producing clones. So this is the method for hybridoma. The main disadvantage here is basically, so whatever antibody you get, these are the chimeric, I could say the mouse antibodies. So mouse antibodies gives lot of, allergic reaction when it is used in therapeutic antibody. That is the main disadvantage. And these days, all the companies, most of the companies are working on a fully human antibody. A fully human antibody basically isolated only by yeast display or a fast display technology. So I will explain you about this. So how MABs are made. So recombinant DNA technology. This is the fast display technology. So in this case, we will have to isolate B cells, antibody producing cells from human peripheral blood. And then there is amplification of variable heavy and variable light chain, or maybe a full antibody. So we will have to design a primers which can amplify very specific antibody genes from the B cells. And then it has to be displayed on a phages. There are different vectors which specifically display a protein, a very specific protein, a single protein on a surface of phages, bacteriophages. That is the reason it is called as the antibody phage display technology. So it is expressing antibody protein or antibody on its surface, and then we will have to select. I could say that once we will make a library, it is immune system outside of your body. If you are making a naive library, and if size is maybe 10 raised to 9 or 10 raised to 10 different clones, so it is immune system outside of your body which is expressing all 10 raised to 9, 10 raised to 10 different clones. You must be aware that the antibody gene, gene rearrangement theory by Tongawa, who has got the Nobel Prize. So different genes can be amplified, expressed, and then we can isolate a different antibody gene or antibody clones from the library, from the naive library. But what is the difference, between naive library and a very specific COVID-19 library? So in that case, maybe we can isolate B cells from a COVID infected and recovered person, a survivor, COVID survivor, so that it has been infected by a virus, COVID virus, and then there is an immune response. The B cells has produces some antibodies against spike proteins or some other part of the protein, but we don't know where it has been, how to, isolate those particular antibodies. So then we will have to make a library, and once library has been done, then there is a enrichment process. So size of the library is maybe 10 raised to 9, 10 raised to 10. So I am working with one of the company. Because of the confidentiality thing, I cannot disclose the name. So already we have filed a provisional patent, and now we are planning, soon we will file a sequence based patent. So work is going in a very good swing, and we soon we will isolate neutralizing antibodies. So then we will publish data, and after filing a patent. So basically, uh, I could say that. So uh, antibody library construction has been done. So what is antibody phage display library? Already I have explained that the VH and VL, variable heavy chain and variable light chain, or full fab, can be cloned in a phagemid vector, and then it has been transformed into E. coli cells, and then it has been expressed on phages, surface of the phages. The result library of the phages expressing antibodies and a specific surface, and there are different types of vectors available in the market. So it depends actually what type of antibody you are expressing, whether it is a Fab or a CFP. So once you have made the library, it is a pool of genes. So n number of antibody genes are there, maybe, and from that you will have to isolate a very specific antibody. In case of COVID, the target is basically spike protein, S1 or S2 or RB. So in that case, what we will have to do, once you have a library in hand, you will have to grow the library, you will have to make the phages, and then you will have to enrich your phages or antibody. Instead of phages, I would say that the antibody producing clones, you will have to enrich those, and that process is called as the biopanning. So once you will enrich it, so you are just isolating a specific phages against your antigen, maybe a COVID antigen or a spike protein S1, S2, and then eliminating the other non-specific antibody clones. So the biopanning process is biopanning process 1, 2, 3, 4. These are the enrichment process. Then you will have to keep doing it maybe till biopanning 3 or biopanning 4 till you get your binders. Once you have done those, then you will have to do an ELISA. So this is the construction of naive library. Already I have explained about the isolation of lymphocyte from donor, isolation of total RNA, and reverse transcription of into cDNA, cloning of antibody gene into a phage display vector and display on a phages. And then the second stage will be a biopanning stage. So in this diagram, you can see that if you have coated a specific antigen and then you are putting your phages, the library phages, and then you are enriching these phages against your particular antigen. So in panning one, maybe you will have a very specific 10% of specific clones. Then in round two, then you will have a 70-60% of your specific clones, and in round three, maybe you will have a 99 or 98% of specific clones. And then after doing ELISA, you know that whatever clones you have isolated, it is producing antibodies against your target antigen. So these are the steps: build a human library, display on bacteriophages M13, biopanning on a very specific antigen, then screening of antibody against particular antigen, any antigen. Then validation of binders. So there are different methods to validate binders, like you have to do a Western blotting, then we will have to do a sequencing of variable heavy, variable light chain, or maybe a full length gene. And then we will have to determine the affinity of that particular molecule, how much affinity it has towards the target. And then that process, you will have to select maybe 100 different antibody clones, and based on the characterization, suppose maybe you can select a 10 different antibody clones, the topmost 10 clones, which has a better sequences, which has a better antibody antigen binding capacity. And then those clones, once you have narrowed down that thing, maybe from 100 to 10, or maybe from 1000 to 50, those clones will enter into a toxicological studies. First is the toxic studies, because any molecule we develop, the safety is the main thing. Efficacy, I could say that efficacy comes later, but it should be safe. Then we will, it goes to a preclinical studies where we test the toxicity of that molecule. Once it is not toxic, then it will go to a efficacy. So then we will have to determine the efficacy of that particular molecule, whether it is a chemical or it is a biological molecule. Then after efficacy in a preclinical trial, then it will go to a phase clinical trials. In phase clinical trials, basically it is on a very less number of participants, maybe 100, 200, as per the guidelines of the different countries, maybe US FDA, or any different body, government body. And then in phase two, just if the results are promising, then it has to be, phase two has to be conducted on a more number of participants, maybe 1,000, 2,000 at a different location. And then the phase three clinical trials, and then molecule comes in the market. So it is really a very lengthy process. Even though you have a molecule, if you suppose you have 10 different molecules, all are better molecules, then at a time you cannot do a preclinical trials and clinical trials of all the molecules, because it is really a very costly affair. Even for one molecule, we will have to, company has to spend maybe 2 to 3 crore rupees in toxicological studies and preclinical trials. So all molecules cannot be tested. You will have to keep the backups, and then if you have selected one or two clones and those clones are not doing well in toxicological studies, or maybe in I could say that in if these are not efficacious, then you will have to test the remaining two best clones, maybe the clone number 3 and clone number 4. So that way, so these are different types of therapeutic antibodies: the mouse antibodies, then the chimeric antibodies, where the variable heavy and variable light chain are from human. Here it is fully mouse part. Then the second generation antibody, I could say this is the third generation, and this is the fourth generation, I could say. So here only the uh, CDR part is from human, and other part is from mouse. Here only here, sorry, FV part, the 5 part is from human, and here only a CDRs are grafted from humans, and then the overall molecule is from mouse. And then this is the fully human antibody. So by phage display technology, that is the merit of the technology, that we can isolate a fully human antibody from this technology. So already I have explained about the validation of binder specific antibody. So ELISA, DNA sequencing, flow cytometry, and the BR studies we will have to do. And as you know that there are two different mechanism how antibody kills the specific antigens or bacteria or viruses, ADC, and then the CDC, the antibody dependent cellular cytotoxicity and the complement dependent cell cytotoxicity. So what are the pros and cons of this? The pros is basically antibodies are very specific and it has very low allergy or no allergy. And large scale production is possible where you can produce antibody by fermentation method. Cons are time consuming. It is really take lot of time and money consuming, and lower affinity as compared to the polyclonal. And once if you are getting a binders which has a low affinity, so by antibody engineering you can just increase the affinity of that particular antibody gene, where you can just you can change the specific amino acid in the CDRs, variable heavy or variable light chain. So different application of antibodies. Examples of some important therapeutic monoclonal antibodies in market already. This is the Avastin. It is used for cancer. The overall market size is 5.7 billion. Rituxan. It is used for rheumatoid arthritis, non-Hodgkin's lymphoma. Market size is 5.6 billion. Likewise, Humira, and it is used for RA. But the current size of this, it is a top-selling brand worldwide, and the current market size is approximately 20 billion US dollar. It is equivalent to Indian domestic market. The Indian domestic size of Indian domestic pharmaceutical market is $20 billion. And then the Remicades, 3.5 billion. So main therapeutic areas we are interested in: inflammatory disease, RA, SLE, and maybe organ transplant, solid tumors, and now working on COVID neutralizing antibodies. So what are the targets in case of COVID-19 neutralizing antibodies? So the targets mainly worldwide, the targets are the same: the S1 and S2, the spike protein 1, spike protein 2, and the RBD. But in this case, once you have the antibody, the advantage is that even you can make a cocktail of two different antibodies. So what is the advantage of making, cocktail antibody? That can be discussed later, or I can explain you now. So basically, the advantage for making a cocktail antibody, if there is some, mutation. So as you know that virus is getting mutated in every country. So if there is mutation in S1 protein and you have produced antibody and formulated it into injectable form, and there is a mutation in S1 protein, then it may not work. It will not bind to a virus. It will not neutralize the virus. So in that case, what can we do as a researcher? We can formulate an antibody, two different antibody, maybe S1 and S2, two different antibody in a single wire. So that if there is a mutation in S1, then the S2 antibody will bind and it will neutralize the virus. So it is a dual effect. If there is a mutation at S2 place, then the S1 antibody will be effective. So that way, it's a double attack on a virus. So that cocktail antibody concept is really good. I think that some companies, they have not disclosed. So already few companies has got the US FDA approval for the treatment of this, deadly disease, this COVID. One is the Eli Lilly antibody. I think this is the first antibody which has got the US FDA approval. Regeneron's antibody also got the approval. And recently, one Asian company, Taichan, Singapore-based, it has started a phase 3 clinical trials. And then this is the structure and how antibody works, basically, the spike protein. So this is the target. So basically, this spike protein, it binds to a receptor. So you can neutralize the virus, or we can just target this particular protein, and then this antibody will bind to this. It will neutralize or kill the virus, so that it will not bind to the receptor. So it will not eventually enter your lungs or other part of, there are receptors in intestine. So it will not basically go and, infect your lungs or intestine. It will be neutralized. Vaccine. So I don't know much about the vaccine. So basically, what are the advantages of, we can, I will just explain you what are the advantages of, antibodies over vaccines. So this Oxford vaccine says that the efficacy of vaccines will be efficacy, or maybe it will work in 62% public worldwide. So what about the remaining, 38% population? Even, some person is getting vaccine, his immune system is not producing antibodies, so that person is prone to infection. He has not got the, his immune system is not, responding to that particular vaccine. So in that case, he is prone, he can get the infection. So mostly the elderly people, maybe the kids. So in that case, once someone has got the infection, this antibody will be used as a, there are two, either it can be used as a prophylaxis like vaccines, or it can be used, after infection also it can be used for the treatment of COVID infections. So how many different antibody candidates? So there are, 67 antibodies program are in discovery and about 51 antibodies program in preclinical trials. 3 or 4 has already got the approval, and some molecules, are in preclinical trials. Some are in, maybe in phase 1, phase 2 clinical trials. So worldwide there are more than, 198, maybe around 200 antibody candidates in different, phases of clinical trials, and some might be in a, discovery program. This is the overall, size of the antibody. Already I have explained about the polyclonal, molecular, monoclonal, nucleic acid. Yes, so thank you. Hello. Hello.
Uh,
I think it is fast.
no, it's
uh, uh, so I appreciate the, any question related to
Thank you.
the need of, discovery, or related to
Absolutely.
antibody fast display platform, or maybe any other question.
sure, sure.
Yes. So thank you, Dr. Kafil. First of all, for the general information of audience, I'll just like to tell that when I spoke to Dr. Kafil last night, he was not well, and he was, he had through a lot of exhaustion, but finally he agreed to come in today. So thank you, sir, so much. In spite
Thank
of
you.
your ill health, you came in and you prepared a lot of slides. So on behalf of our faculty and everybody around here, really like to thank your time and efforts in coming in, really enlightening us, because everybody is talking about these areas about antibody development.
Yeah.
So the first question I would like to throw in essentially,
Yeah.
and then I can see a lot of questions coming in here. That essentially you've talked about, there's a lot of talk about mutations. There's a lot of talk about the virus being mutated essentially. So when we talk about antibodies essentially, and when we think of therapeutic antibodies, so doing a purely monoclonal to a very specific target would be helping, or doing a cocktail essentially, as you mentioned essentially. What is the preferred pathway that the companies are taking in essentially? That's one question. And the second question was mostly related to vaccine development. So, and if you think about it essentially, in terms of the elderly patient population essentially,
Right.
the response to the vaccine essentially in terms of a immune response, how do you think that pans out well for them to give an effective protection against the virus essentially?
So basically, first thing is the development of, antibodies. So cocktail is definitely a better thing to make, because it is, it has a double protection, and this virus is getting mutated, as you know that. I mean, IGIB has done a sequencing of, virus, COVID-19 viruses from Indian population. I mean, from different part of India, and they say that there are different, maybe around 1,000 strains are present. I mean, there are little mutations. So that way it is going definitely, better to make a molecule which is a cocktail molecule. And as far as I know, they have not disclosed whether it is a cocktail antibody or a single antibody, because these big companies, they don't want to disclose what type of formulation they have done. But there are several, couple of papers which suggest that, it is better to make a cocktail antibody. So it is, I mean, it is not a bispecific antibody. Either you can have a two different arm, antibody arm, SCFV against S1 and then SCFV against S2. So likewise, this arm can bind to S1 and then S2, or you can make antibody which has, these two arms bind to only one antigen and then another antibody which binds to S2. And then while formulation, maybe you can have a 50-50%, combination of that, and then you can formulate it. And then the
Yes,
second,
yes. Yes, sir. Go ahead.
uh,
Yes.
uh, and the second part, the doses, because everybody is, as you know that, I mean, whatever I know, I'm not a vaccine man. I don't know much about the vaccine, but as far as I know, I mean, there is not, any vaccine in the world doesn't have 100% protection, maybe a 60 or 70% or 80%. So what about the remaining population? So these antibodies can be used as a prophylaxis. So these are the ready antibodies even before infection. Those who are, high risk group, maybe the doctors and the healthcare professional, they get the antibody doses before getting the infection. So that ready antibodies against COVID-19 will circulate in the body, in the blood, and then whenever there is infection, it can fight to to those viruses. So that is the thing.
Okay, great. Professor Madhuri is asking in terms of is there any conventional dosage forms of delivering antibodies?
I think the dosage is approximately 300 microgram, maybe a weekly doses or a monthly doses. I don't know much about the formulation. I mean, the dosage form, because we are busy in, just making antibody, and also whatever reading we are doing, we are just, reading. I mean, just how to make it, and know how to make a better molecule, efficacious molecule that will come at a later stage. So, but it is intravenous, and the dosage is 300 microgram as far as I know, and for maybe 2 or 3 doses. So it will not fade immediately. It will be in your body maybe for 4 to 5 months or 6 months. So once you have taken an injection of antibody, it will circulate in your blood for 4 or 5 months.
Great, thanks. I request the faculties to post in their questions, and if they want to directly unmute themselves and ask it, they are free to do so.
Yeah, I think I just make it more interactive, instead of, just I was walking through the slide and then no one asked the question. So you can ask, anybody can ask any question related to antibody.
Great.
So fortunately,
Yes, yes,
before this antibody thing, it was mainly for autoimmune. But after this COVID, I mean, it is in demand. This technology is in demand because now this has been diversified towards the infectious diseases. I mean, now
right.
COVID, maybe later on, maybe tuberculosis and some other diseases where you have a specific target and you can make antibody and you can just kill that particular antigen.
Right. So I think what you're saying is key because of this whole pandemic, there has been
Yeah,
accelerated technology development, and that
right,
would
right.
benefit other disease as well,
Yeah,
essentially.
yeah. Even there is one antibody, anti-VEGF antibody that is used for age-related macular degeneration, AGM.
Right.
One antibody for
Right.
RSV. This is again a viral disease, so it has been discovered and already in a market. So this, I mean, this field overall has been, diversified into infectious disease research and development of some therapeutic antibodies towards it.
Yes, I think that will be very
Yeah,
useful, especially for diseases such as tuberculosis, which are the poor man's
yeah.
disease. So,
Because I mean, because of the MDR and the XDR, so antibiotics are not working for the mycobacterium.
right. Uh, Professor Majid is asking in terms of person given antibodies prophylactic like healthcare professionals, will it prevent the effectiveness of vaccine to induce immune response in those people?
I don't think so. Oh, I mean, yeah, I don't think that it will prevent, because that is a ready antibody injection. It will circulate. Even someone will get the vaccines, then their immune system will also produce some antibodies, because those antibodies and this antibody, these are the engineered antibodies, and it may be a different antibodies what that person will produce after injecting the antigen. So I
Yes.
think, though I don't think that the person who has taken the antibody cannot take even the injection of vaccine. So they can, I mean, that person can take the injection.
So just as
of
a naive
vaccine.
question, Dr. Kafil, when you read in literature, and this is coming from someone person who's not very familiar about antibodies production in the body, they say that let's say a person is infected and there is a whole array of antibodies are being expressed, and they say some of them, most of them are not very useful, only
Right.
some are neutralizing. So what does that mean essentially from a from a scientist perspective or
Okay.
biotechnology perspective? Can you
Yeah.
expand on that?
So yeah. So I mean, I know that. I mean, you must have heard about the antibody gene diversity. There is a different RJDC, VDJC gene rearrangement. So at a given time, everybody has approximately 10 raised to 9 or 10 raised to 10, maybe 10 raised to 9 means 1 billion or 10 billion different antibody genes. And when there is infection of antigen, a specific antigen, that type of gene rearrangement will happen. And then your immune system, the B cells, produces antibodies, specific antibodies towards that particular antigen. So there are
Right.
some strong antibodies, maybe the weak antibodies. So that particular antibody isolation, maybe your set of antibody gene is different, my set of antibody gene is different, and maybe Dr. Abu Sufyan's set of antibody gene is different, because since your childhood you have been exposed to different antigen, you have been exposed to different antigens. Accordingly, your pool of antibody pool is different. Mine antibody pool is different, and when somebody's antibody pool is different. So when we make a library, we will try to make a library from maybe 100 different individuals, so that we will have a highly diverse antibody pool, so that there are high chances of hit rate will be more, and then we will get more number of better antibodies or specific antibodies. So
Okay.
that way, because what is happening in this case, in case of COVID, there is a T cell response as well as a B cell response. So
Right. Right.
in some cases, in maybe 50% or 60% cases, there is a production of antibody. But I mean, so far we know that there is a production of antibody, but we don't know who has produced a better antibody. There is no method to determine that. Suppose a 50 person has got the infection, and their antibody has been, their immune system has been produced antibodies, and then they have the antibody. We know that they have the antibody, but we do not know. There is no method to detect or determine that which person has a better antibodies.
Okay. Okay.
That that pool, we will have to make a antibody library from different persons, and then we will have to isolate antibodies. Even we suppose we have 100 different clones that will neutralize the antibody. These are the ELISA positive clones. The affinity of antibody will be different, and then you will have to select we will have to select the best affinity binder, so that it will immediately bind to the virus and it will effectively kill the virus.
Utilize it. So you're saying that if I say if I as a patient am expressing neutralizing antibody, that would be different from someone, let's say in a Chinese population or in a Caucasian
Right, right.
population. There is
Not
a chances that
yeah,
it would be
even
different.
from a same Indian population. Yeah,
Okay.
yeah.
Okay. Great. There are a lot of questions coming in. Professor Jayashree is asking if for the COVID vaccine, if the spike proteins are targeted to avoid entering host, but what if the virus is already infected the human body? So then the use of antibody and let's say vaccine, she's asking, then what is the value of that means essentially?
uh, Vaccine might not work if already has someone has got the infection. So in that case, then we can, I mean, physicians can use the antibody, because these are
Okay.
the readily available
Really made.
antibody. Once you inject, it will bind to the target, and it will probably kill the or neutralize the virus.
Okay.
Because vaccine response will take some time, I think
Fine.
2 weeks, maybe 1 or 2 weeks or 3 weeks, and once you get a
Right.
booster dose, then antibodies will develop in your body.
Right. Right. Professor Arul Selvan is asking in terms of cocktail antibodies are not going to alter autoimmune system of the individual over a period of time. Altering autoimmune system is safe, or it's going to create some same effect like a nanoparticle or so.
I mean, I don't think so. But once this antibody will come in the market already. I mean, two antibodies are in the market, and then we will do a post-market studies, maybe after 6 months or 7 months. Then we will come to know about what are the main side effects. Because even in phase clinical trials, we conduct a clinical trials on maybe 200, 2,000, 3,000 people. But when it will go in a market, it may be useful for thousand, I mean, crores of patients, maybe 1 crore, 2 crore, 10 crore people. So once we will have those data, the pharmacovigilance will collect the data, and afterwards, they will evaluate it. So I mean, it is really difficult to say right now what would be the
Right.
eventual side effect when we will use it on a mass population.
Absolutely. I
Uh,
mean, I think we are
uh,
seeing that with the Pfizer
yeah.
vaccine that is going on. That people are
Right.
having some anaphylactic reactions to that.
Yes.
I think that evolves as you do vaccination in mass population.
Yeah. So
So
yeah, I mean, as you know that any drug doesn't matter it is a
any
chemical or
drug,
a
absolutely,
biological, it has a few side effects. Yeah, yeah, it has a.
absolutely,
But
absolutely. And you
right now we
uncover
need
that only when you
some.
do. Yes.
Yeah.
Professor Darshna Matra is asking: is cocktail antibodies can change any functional systems in human being? What kind of different formulations can be formulated to deliver these antibodies? I think we spoke about formulation a little bit.
As you know that antibodies are very specific in nature. It will not basically a monoclonal antibodies. It will not even bind to any other antigen which has a difference of even one or two amino acids. See, because even though there is a
Hmm.
difference of one amino acid in a protein, it will change the overall structure. It is not one amino acid. Even there is one amino acid. So as it will bind specifically to a S1 and S2 protein, so it is not present in our body. It is not even present. The similar type of antigen is not present in any other virus particles, spike protein. So it is
Hmm.
very, very highly specific. I think that even the MERS virus is, related to COVID virus. But
Yes.
the spike protein structure is different. The amino acid structure is different.
Yes.
The sequence is different. So even it may not work for the MERS as well. We will have to develop antibody for different proteins or spike protein or any other protein for MERS. So even as far as I know, even there is a difference of one or two amino acid, it will not bind to any other antigen. So
Yes. So
highly, highly specific. Yeah.
so going back to that, I mean, there is what the literature says and what people are looking at is that luckily the virus is not mutating as sporadically as, let's say, other viruses have been shown to mutate. Most likely because of
Yeah,
mutating
apparently, which
mechanism.
is a good news.
Apparently, is
Huh?
a good
Yeah.
news. But let's
Yeah.
say it does, and
Uh,
then the whole thing, the whole process of the vaccines being developed and the antibodies, obviously, then that comes into question, right?
Yeah, but even the vaccine, they have VLP virus-like particles, and then they have also a cocktail. So maybe a S1, S2
Mm-hmm.
antigens, and then they will formulate, and then they will inject, so that there is a response of from your immune system from against both the antigens. So that way, I mean, whatever I have discussed and understood from my friends who are working in vaccine industry, so I had asked them this type of question. So they said
Mm-hmm.
that
This is
different programs, different mRNA vaccines, and maybe a cocktail type of antigen. So they have not disclosed their technology details
right. Right.
because it can be copied by other companies. So that is the reason they have not disclosed these things. They might have
Right.
disclosed to the government agencies, but not, it is not in public domain.
Public domain. Okay.
Mm.
Okay.
So,
So faculties can continue to post questions. I think it's a very interesting session, and we rarely get people who are actually working on antibodies to come and address our questions. So, Dr. Kafil, in terms of a bigger philosophical and a larger question as such, now we are going into this whole mass vaccination drives, and across the world. I mean, US is already, UK has already started. US, they've got emergency approval yesterday. So
yeah,
when you see a vaccination program start, I mean, obviously the most vulnerable among them, the older population, the
right,
effectiveness of vaccines is still in question essentially. So as I
right,
understand, what you're doing now with the mass vaccination is creating what kind of is something like a herd immunity, I guess, right?
right.
So is that, I mean, how do you see that essentially in terms of slowing down the whole spread of COVID within the communities as such? I mean, it'll still, you think it'll still take time or a month or so to, after the mass vaccination are done, to taper down those numbers essentially?
I think that it is very difficult to predict. I mean, maybe the epidemiologists may answer this question properly, that what type of after vaccination, what would be the response and all. So
Hmm.
I don't know, but I think it is better to have two options, two better molecules which can fight this
Right.
deadly
Right.
antigen, I mean the virus. So we have one hand we have a vaccine, and one hand we have antibody. It is
Mhm.
better that we have two different targets, two different therapeutic molecules. So some population, maybe half of the population or more than half of the population, vaccine is useful. Maybe for half of the population, less than half of the population, antibody will be useful. So
So
that way, we have two different
two different.
therapeutic molecules.
So
It
when
is
so
I don't want to say that there is a competition, but because
right.
both would be useful.
So I mean, from a layman's point of view, let's say you have the Moderna vaccine coming in, the Pfizer vaccine is coming in the market. So in terms of giving it to which population, which vaccine, I mean, how does the decision making will happen at the government level? And you as a scientist, what would your
I mean,
thought be?
I really do not know. So I don't know much about the
No, let's
vaccines.
say if you have two multiple vaccines coming in, so I mean, then will people get prioritized to one vaccine for the other? Is there a scientific rationale to that? I mean, just a naive thought I have in mind.
Mm, I really do not know. I mean,
Right.
I may have
Because
to
there's
ask
so
this
many
question.
of vaccines coming in the
Right,
market, and there's so many in
right.
pipeline in phase 3. So I'm just
Uh-huh.
visualizing.
As far as I know, I think that the vaccine which has been developed by Oxford and then it has a joint venture with the Pfizer, and finally it is manufactured in SII facility, Serum Institute of India. I think that would be a better vaccine. So and
Okay.
it say that it will give protection to 60% to a 90% population. So
Okay.
I personally, I mean, this is my personal opinion. So
Right. I mean, looking at the entire push for the whole vaccine development, I think the similar push should have been done for the antibody development as well. I think because if you
right,
do a dual approach, essentially, especially for
yeah,
people whose systems are not capable enough to elicit that response, for
response,
them, antibody would be a better choice to start
yeah,
with, essentially, right?
yeah.
Where do you see the whole costing thing coming in, Dr. Saab, here, essentially, with the antibodies? I mean, we have this whole scary history of we importing antibodies from US and others, oncology in our area, hugely expensive. People like it,
Right,
or most of it is out of pocket, it becomes very restrictive. So,
right.
what?
In case of this biosimilars, the price has come down. Now the antibody injection before even Herceptin, the costing of Herceptin for 400 milligram was more than 1 lakh rupees. Now it is 22,000. But for a therapeutic, it will be less because the dosage form is just 300, 400 microgram. So
Okay,
it might be a slightly costlier than vaccine. Definitely, it might be, but I do not know the price of antibodies. What price they are giving at US, because so far I think these antibodies are not available in Indian market.
right.
So if we develop, I mean, success, if we or any other Indian company will develop an antibody, it will be really good for the. I mean, this is the good time to serve the community, and it is really a boon for the patient worldwide
Hmm.
if
Hmm.
we will be able to develop some novel antibodies which can neutralize the virus. So it
Absolutely.
will be really a boon for the patients globally,
Yes, the
and
domestic.
it will be and then the definitely if we manufacture in India, the cost will come down. So
Absolutely.
that way,
And especially that is very important for the
yes,
rest of
yes,
the
the
global
affordability,
population,
yeah,
especially in the eastern
affordability, yeah,
countries and the African
yeah,
countries who
mainly for the developing nations.
yeah,
So
exactly,
yeah,
for the
so
developing
cost
nations.
cost is a matter. Yeah, is a big,
Cost is a big matter, essentially.
yeah, affordable. It should be affordable.
Absolutely.
There might be some government there might be some government programs or maybe assistance, or they will buy antibody or vaccines, and so that
Right.
way it might be there in their budget, so
Right.
and it should be.
It should be, obviously. Questions coming in. There are no questions in chat. Any other questions, essentially? Uh, the floor is still open. Dr. Kafil is still here, essentially. Any questions that anyone would like to post in, they can post. If they want to unmute themselves, they can do that. Uh, any changes? Professor Nilesh Kutli is asking any chances of monoclonal antibody for COVID? I think that's what Dr. Kafil and his team and others are working on, I guess, right?
Yeah, yeah, there are some other companies as well, but yet no one has published any paper or some data. Maybe there might be some other companies, but very, I mean, limited or less number of companies are working on development of neutralizing antibody in India, as far as I know.
Okay, and none of them are in
But
clinical trials, I guess, right?
no, not
None
in
of them.
even phase one. But as far as I know, that Serum Institute has tie up with some company for manufacturing, like vaccine. Even
Yeah,
this
yeah, that's Outslash Institute.
vaccine, yeah. The same way they will do a joint venture or some collaboration,
Yes, yes,
so
yes.
that they can just do a
Mass
manufacturing.
produce it.
Yeah, just the production, not the research.
Yes, just this production. Same with Bharat Institute, essentially.
Yeah, same way SII and the Bharat. So
Yeah.
that way.
So nothing internal as such.
Yeah.
Yeah. Okay, so no paper. Great. Any other questions before we uh, say goodbye to Dr. Kafil out here? Last minute, no questions? Okay, great. Professor Abu Sufyan, it's over to you.
Okay, thank you. Thank you, Dr. Kafil, for your very informative session on antibody for COVID-19, learning from product development. You have given wonderful insight on antibody, step in its development, and also very interactive question and answer session. Going ahead, today we have a next session on journey of product to therapy. For this session, we have with us Dr. Rana Reis. I take this opportunity to introduce her. Dr. Reis received her PhD in pharmaceutical sciences with substantial experience in biopharmaceutics, drug permeability, drug transport, drug metabolism, pharmacokinetics, and drug delivery, including product design and characterization. She served as the director of the Drug Metabolism, Pharmacokinetics, and Bioanalysis Laboratory within John Hopkins Drug Discovery. She has over 15 years of experience in designing and conducting all DMPK and drug delivery strategies using appropriate in vitro, in vivo, and in silico methods impacting drug discovery projects. She has also served as DMPK and drug delivery consultant to several pharmaceutical companies. She has published over 80 manuscripts in high impact journals, including a recent landmark publication in Science. She is co-inventor of over 14 JHU patents, 6 of which have been out-licensed. She has also co-founded 2 JHU startup companies, which have amassed over $40 million in external funding. Thank you, Dr. Reis, for joining us. I extend my warm welcome to Dr. Reis and request you to start with your session. Dr. Reis, to you.
Good morning, everyone. I mean, good evening, everyone. This is early morning here in the United States. Thank you, Abu Sufyan, for a nice introduction. And thank you to Dr. Said for inviting me to present today. I need to share my screen, so I think you guys will have to give me the permission to do that.
Dr. Sharik, you need to give the permission.
Yes, give me 1 second. I'll do that. I'll make her the co-host. Sorry about that. A very good morning to you, Dr. Rana Reis. It's really a pleasure to see you here. Thank you so much for joining in early morning. I can imagine that. Okay, I think I've given you the rights. Please check at your end. Are you able to present?
All right, thank you. So can you all see my screen?
Absolutely, yes.
Clear. And can you all hear me clearly?
Fantastic. Yes, very much.
All right. So great. So today I'm going to give you actually a pretty interesting story of a compound, which is a glutamine antagonist compound. And the stories from really bench to bedside. So this compound is exciting because it was actually developed in my lab. And beyond that, we discovered this compound, tested this compound, and now this compound is in actually phase 1 clinical trials, and it has been given by FDA fast track to take into lung cancer. So I thought this would be a very interesting story for all of you to go over. So I'll get right into my talk. Today, before I get into the science of glutamine antagonist prodrugs that we have developed in our lab, I would like to first give you a brief intro to who we are, what we do at Johns Hopkins. Because we are a slightly different group, being in academia and serving as a big drug discovery center here. And so just wanted to give you an idea of what happens for a drug discovery center, how we function and all, and then go into the discovery of glutamine antagonist program, revamping it actually, because this was a compound which was developed long time ago or tested long time ago, but was not developed. So slightly easier because we were not doing the chemistry and everything here, just doing the prodrug and drug delivery. But the thing that the pharma looks for is, of course, the robustness of the data coming from academia is important. It should be reproducible. And the first thing that every pharma that we've worked with or whoever has bid on our compounds is they like to repeat whatever we do. So you have to make sure that what you do is should be reproducible, should be accurate, should be precise, and that's what science is all about. So we have, like I said, we have the group we have is mostly from pharma, so they have some idea of what is required by pharma. So, and we work according to that. But in general, I think the one thing, and there are multiple papers on this too, that while academic drug discovery or science in academia is very exciting, a lot of it is sometimes not reproducible. And I think making sure that the data that is being generated and is well validated and confirmed before you present to anyone, I think that's very important.
Yeah, I think you rightly pointed out essentially. So the way, and a lot of time academia approach is mostly into kind of, building up the research, more into publications, but I think the industry approaches from a very different way. They want to see validation, right. They want to see reproducibility. They really want to pressure test it before taking forward, because there are a lot of things at stake. Professor Salman, he talks about on a similar tone. He says, what's the difference between doing research in industry versus research in an academia, essentially?
I think so. The problem, I'll talk both good and bad, right? So in academia, like I said, it all looks like, we are all perfect. We are at Hopkins, we get access to everything, but it's not easy, because not only are we doing all of this drug discovery, but we are also like, I have a dual appointment. I'm not only the director of drug metabolism, pharmacokinetics, but I'm also an associate professor in neurology and now recently in pharmacology, which means I have to maintain both, right? I have to do this drug discovery and do it well and think about all of this. But at the same time, I'm mentoring postdocs, I'm mentoring students, I'm teaching some classes, not 100% because neurology doesn't require that, but I have to get grants constantly. And we at Hopkins is all soft money. So we are 100% funded by our own salaries. So for that, we need constant funding and constant grant money and foundation money, or we have an IBD project. We are constantly funded by Crohn's and Colitis Foundation. We get sponsored research, even though, so whenever we give our compounds to pharma, we still get SRAs from them. So we, like, for doing these backup compounds and all that I was talking to you about, that's also one way to fund. But 80% of our funding comes from NIH grants. So we write these drug discovery grants to NIH, and we get R01s or we get U19s, the big grants, so that we can, again, move this forward. So unlike pharma, where it is all funded, all you're doing is moving the compounds forward, we have to do both. And being in academia, we have to constantly publish, and that's why we are big into publication too. So, so if you have to keep up the best of both the worlds, both academia and industry, when you're an academic drug discovery group. I feel like I've become a jack of all and master of none, maybe, but it's all for good.
Absolutely, I can imagine it. It's all about balancing both of it, and it can be challenging. I mean, academia,
it is very
can
challenging.
be very pressure testing, especially the continuous pressure of bringing in funds and the whole cycle of continuation at the academia. It can be stressful. I can imagine that.
Yeah,
so,
stressful, but nice way. You get
nice.
Good and bad anywhere, right? So it's not that bad.
I think this is the fun of working in academia. Is all about interesting people, and there's a little more less stressful environment compared to industry. You get to meet a lot of interesting students, postdoc. I think that also keeps you a little.
And also you get to work on what you like to do. So I think the biggest thing is we select our projects.
Right.
They are not designated to us. So if we like something, we work on that. And that's why it becomes very interesting for us. So it's not something like, and the second thing is, even though like in pharma, if something doesn't work, it's like scrapped off and you don't work on it anymore. But in academia, we still try to get publications, grants, and things out of it. So try to make the best of where we are.
Absolutely, absolutely. Salman has a follow-up question. He says, how challenging it is to get funding from industry, joint ventures to prove a project in terms of translational research, essentially.
No, it's not easy. So you have to, like I said, you have to put in a lot of work, and being a scientist and selling the project is two different things, right? So one thing is your science is good, so you can say all of this is good, but what I've realized is it is not just the science, it's what you present and how you present, and a balance of what they're trying to hear from you is and not going down to the level of, oh, this specific gene was doing something. It's more about what's a big picture. How many patient population is there when pharma is looking at something, and or what will be the target, what countries are you targeting, and things like that. So they are looking mostly at the selling strategies of the compounds, and we sometimes get hung up on the science. So it's not easy. It's pretty challenging to get VC money, but once you build a repo, you show that you've been successful in doing this, and then you just attract more money like that. But I think the beginning part or the starting part is always the hardest part.
Yes, I can imagine that. I mean, it's just a different approach of we're looking at things. I mean, industry looks at it from a very different perspective. Sometimes in academia, you're purely looking at science, and that really doesn't work. I still remember my early days at BMS, and we were working on a project and a very impressive compound in early phase. And I was a part of that drug development team. And lo and behold, we really seen some very fantastic results, preclinical results, and the team decided to just kill the compound. And it was a shocker for me, essentially. But then say, Pfizer has a compound which 6 months ahead of us. We don't want to be second in the game. So, and that that's the kind of a shocker that you get, essentially. You've mentioned, starting two companies. So how was that experience? I mean, being, launching two companies from an academia lab, how cool it is, how exciting it is, how challenging it is. And a lot of people in India also, they think about, incubation. You talk about, setting up companies as such. What's your experience on that?
I think, again, like I said, nothing is easy. So when we discover compounds or we have an asset, all of our inventions, they go through Hopkins. So we have two options, right? Either the Hopkins Tech Venture Group, which is the JHTV, Johns Hopkins Tech Venture Group, they take the compound and they try to sell it. Or the other way is to first spin out. Mm-hmm. And, for the spin out, we need to take all of our inventions into the company and buy them as founders. And then we try to sell the company, the company that we find. So in the case of founding the two companies that we did, it takes up a lot of takes us out from science, and we have to make sure that then we are the ones responsible for bringing in the funding. We are the ones responsible for doing all the due diligence. We are the ones responsible for backing up our compounds and things like that. The good thing about that is that because we are the ones negotiating, we try to do the best we can negotiating wise and trying to get as much money as possible into the company, or and do it on our terms. But for that, we need some initial funding money because we have to buy our own inventions, the compounds, like for example, the patents we've invented, we have to buy them from Hopkins and bring them into our new curve. And when if we see a smaller asset or a single asset, like not like the 300 compounds I showed you, and we have 5 different patents on this glutamine antagonist program, but we also have some smaller projects where we have a couple of assets in that, like only few molecules that we've made, but they're looking very, very good. So in that case, we do not spin out a company. And the example of that is we recently did a collaboration with or an option agreement with the company in India. It's called Sun Pharma, Spark Bio. And in that case, what we've done is we have this one invention that we are working on with them, and they've taken it and they have repeated everything we've done. And then eventually they'll develop this compound and we will get royalties and things like that once a compound is developed and marketed.
Great, thanks. I think we'll just take this last question. Professor Mirza Anwar is talking about how challenging is industry-academia collaboration. I think you've spoken a lot about that. Would you want to add something to that?
Once you have the collaboration, it's not that challenging. I think again again depends on your experience and everything that you're doing. But when we do the collaboration with industry, we do it on our own terms, and generally the industry would like for us to work on things they think we can do. So if my lab is doing, for example, these CESO knockout mice are not available to a lot of people. So if my lab has them, we are able to do all the pharmacokinetic studies with the prodrugs, which are owned, like Tracen Pharma is not able to do, or other industries are not able to do. So and a lot of bioanalytical methods also are developed in our lab, and we transfer them and everything. It's once you build that, again, you build that collaboration, you build that repo, you're able to work with them. And what I've realized in these last 10 years is, I mean, we are all scientists. We all have the same goal at the end, to get the right results and to be able to see if we get anything positive. So as long as you're confident, as long as you know what you're doing and you're learning and reading constantly and building it off of the right, path and knowing exactly where you're going with it, I think you should be okay. But it all comes with experience. I'm not trying to make it sound easy. I think struggles are always there, but eventually, once you've done it once or twice and you know what exactly needs to be done, you can do it.
Right. Very nicely. So thank you once again very much, Dr. Rana Reis, for it's a very nice presentation in terms of a lot of learnings that we had in terms of science, in terms of your experiences in developing compounds, out licensing them, and I'm very happy to see as one of my colleagues, doing something very fantastic, and very happy for all the success that you have. And we, all of us, wish and pray for you that, you continue to grow in your role and you continue to come over and speak to us essentially at this forum about your new exciting ventures as such. So with that, essentially, I would hand it over to Professor Abu Sufyan for taking this forward. Professor Abu Sufyan.
All right. Thank you, everyone.
Thank you, sir.
Thank you, Dr. Saeed.
Thank you, Dr. Reis, for your informative session on journey of product to therapy. You have given wonderful insight on product development journey, how you are collaborating with experts from different domains to translate product into therapy. You have shared nice real experience of John Hopkins in product development and many more. Once again, I thank you, Dr. Reis, for an excellent and engaging session. Today, we have successfully completed 1 week AICTE-ISTE sponsored faculty induction program on multidisciplinary and translational drug development. Now, before proceeding towards a short valedictory program, although you are giving proper feedback through Google form, but I also request you to give feedback of entire program in one word by using link posted in the chat box. Your feedback shall be displayed on the screen as a word cloud. Let me share the screen so the feedback can be displayed. Thank you. So meanwhile, all participants give their feedback. We had participants from across the country. Some of the participants are Dr. Pradeep Kumar. He's the assistant professor at Kelly College of Pharmacy, Hubli, Karnataka. I request Dr. Pradeep Kumar to please come on a screen and please share the feedback of this 6-day refresher program. Dr. Pradeep.
Yes, sir. Am I audible, sir?
Yes, you are audible.
Yeah. At the outset, It gives a great pleasure to share my feedback on this 6-day AICTE-ISTE sponsored FIP. So in these 6 days, the information shared by the renowned speakers was very informative. So they touched upon many areas of the research, including the computer drug design, then drug transporters, biomarkers and drug development, PKPD models, application of 3D printing in drug delivery technology, then the modeling and simulation role in early stage of drug development. And just now the talk got over by Madam Rana Rice, product to therapy. All the speakers gave their experience in the form of an information which is very valuable for all of us to take up the research to a better height. So altogether, it was a very good FIP conducted by AIKTC in order to bring up some that is better inputs into the research work along with the academics for all the faculties over here. So really felt very happy by attending this, and I wish to thank the organizers who brought all these renowned speakers under a common umbrella, who shared with us the very informative things. So I am really happy, and the timing was also very good. Soon after our college work, we can join. So it was from 5 to 7:30 or up to 8 o'clock sometimes. A very convenient time, because during the college hours means again there will be some works. It may be associated with the inspection or the regular online classes where we can't give our 100%, but the timing was very well that is scheduled. So we really enjoyed it, and I thank all the organizers and the speakers for conducting this nice FIP. And I will be very much happy to again be a part of such programs if at all you are conducting in the future. Thank you very much, sir. Thank you, all the organizers.
Sure, sure. Thank you, Dr. Pradeep, for giving your valuable feedback. Going ahead, we have one more participant, Dr. Priya Shivaji Rao. She is from BN College of Pharmacy, Udaipur, Rajasthan. It will be great, Dr. Priya, if you come on the screen and share your experience.
Yes, sir. Am I audible to all of you?
Yes, you are absolutely audible.
Very good evening, sir, and I am extremely thankful to all of you for organizing this faculty induction program, and it was really a very excellent experience for me for attending for almost all the days, that is 6 days, as well as each and every speaker, whatever they have said, they have cleared each and every questions, whatever was been asked by the each and every faculty over there. And what I actually really liked was they have explained practical concepts, what was really required to a faculty to take that experiences from that speakers and to think on that experience and to prepare such a kind of faculty program in our college, how we can utilize the development skills as well as the development, as well as now just last lecture, that was a very awesome lecture that has explained us regarding the collaborations, regarding the Sun Pharma and all that industries experience. That was a very good experience, and I really thank all the organizers for organizing this FDP at a particular time schedule. As already before my experience, one more sir had already said that the time schedule what we have kept for our faculty development program, that was faculty induction program, that was really very good, that that we get an opportunity to attend properly apart from our daily routine colleges and meetings and all that. So I am really very much thankful to all the organizers as well as the speakers for clearing each and every concept what they want to say to us. Sir, I am not going to tell each and every speaker he has taken what topics that we already have got into our brains and our minds. So we are going to utilize all these skills and all the knowledge that they have given us. And I am extremely thankful to all of you actually for organizing such an awesome faculty induction program. And I really wish all the best to all of you to organize such more events in your college. And we will be very much grateful, sir, for you to be a part of your this kind of program. So once again, I just wanted to say to everyone, thank you very much. And as well as apart from attending this FIP, I would like to thank all the participants also, because if we are having a number of participants, we are very much eager. What is this now? Should also we be a part of that? So I would like to thank all the participants also, as well as organizers, and that is heartfelt thankful to all the organizers itself, and thank you to all the speakers as well. And thank you for giving me such a kind of opportunity to share my experience with you all. Sir, thank you. Thank you so much.
Thank you, Dr. Priya Bansode, for sharing your experience. We found you one of the sincere participant that whenever I start my waiting room, and you were first to join in for most of the time. So thank you very much for being the active participant of this SIP and for your kind information. Maybe in the coming summer, we'll be coming with another refreshment program, and we'll surely share that link with all of you so that you can join the follow-up program to this refresher program. So thank you, Dr. Priya. Welcome. So now we can see here most of them, they have also given the feedback in one word like excellent, informative, nice, and interesting, engaging, great, enlightening. So there are many action verb you have used to describe this the feedback, to give the feedback of this program. In addition to this, if you would like to give some critical feedback, you are also free to give that. You can share that in the Google form feedback link. So thank you very much. Is there anyone else who would like to share their feedback or experience on the screen? You are free to share it. You can unmute and you can share your feedback.
Hello, am I audible?
Yes, you are audible. Okay. You are Ravindra Karande, right?
Yes, sir.
Okay. Thank you. So he is our own student from founding batch, and now he is working in one of the well-known college. So yes, Ravindra.
Okay, thank you so much, sir. First of all, I wish you all a very great evening. As sir told, I am Ravindra Karande from St. John Institute, Palghar, and also a former student of AICTE-ISTE School of Pharmacy, first batch. So I'm very excited to give a feedback about this program. And I would say that it was a very well organized program, and I found it very beneficial and helpful. What I feel that it is beneficial and helpful for all group of people, be it a researcher, be it a businessman, student, or even the academicians like us. This program is a kind of very appropriate way actually to flourish and enhance our network and awareness of different people all over the world. I thoroughly enjoyed all the parts of this program, and most of the presentations were so engaging.
Ravindra, why don't you unmute your video? Let us see.
Can you see me, sir?
Of course, I can see, I can recognize.
Welcome, Ravi. Welcome.
Thank you, sir. Thank you. So yeah, I was saying that the presentations were so engaging, were very beneficial. For me, that this program has generated a lot of ideas about how I can continue to expand my skills, how I can enhance my professional qualifications. I felt like it has helped me to get out of my roots. And I love that the theme was so embraced the conceptual and a theoretical concept and its empirical work. I also like the opportunity of interacting during the session actually. It was a really good experience for me personally, and although the program has ended, but I will always say that it will be remembered due to the way it was conducted, as it was very organized, well organized, good scale, solid content in the presentation. And I do believe that everyone who has attended this has been satisfied with the outcome of this program. And that was only possible because of the organizers who have put their best to organize in the best possible manner. And I'm always impressed with the consistency, commitment, and the efficacy of all those people who have planned this program. And I thank you so much for you guys' efforts for such a wonderful, informative, enlightening program and making it one of the most memorable research activities. So I'm also looking forward for such participation in the events and the activities that will come from the AIKTC. So thank you so much, all of you. Thank you.
Okay. Thank you, Ravindra, for sharing your valuable feedback, and also nice to see you after a long time.
Yes.
Okay. So
Good year.
welcome, welcome. Shall we proceed to valedictory, Dr. Sharik?
Yes, Professor Sufyan, we can move ahead.
Okay, okay. And now we'll initiate valedictory program. For valedictory, we are fortunate to have with us invited chief guest Professor Vijay D. Vaidya, who is Executive Secretary of ISTE, the Director of Anjuman-I-Islam Kalsika Technical Campus, Dr. Abdul Razak Hanutagi, the Dean School of Pharmacy, Dr. Sharik Sayyed, faculty members, and all participants from different prestigious colleges across the India. I extend my warm welcome to all of you for the valedictory of this 1 week AICTE-ISTE refresher program. It's great honor for all of us to have invited guests for today's program, Professor Vijay D. Vaidya, Executive Secretary of ISTE. I take this opportunity to introduce Professor Vaidya. Professor Vijay D. Vaidya has obtained his BTech degree in Civil Engineering from Karnataka Regional Engineering College, presently known as NIT Surathkal in 1981. He worked in the field with MS Afcons Mumbai at different sites in Karnataka, Andhra Pradesh, Goa, MP, etc. He also worked with designer firm and did structural designing for different type of structures like residential, commercial, industrial, and public building. He joined teaching field in 1985 as a lecturer in civil engineering department in Government Polytechnic. Since then, he worked in different capacities, including lecturer in civil engineering, head of civil engineering, training and placement officer, vice principal, and principal. He has worked in administration in Directorate of Technical Education, Mumbai, as assistant director, work as deputy secretary in Maharashtra State Board of Technical Education, Mumbai, which deal with the diploma education in Maharashtra. He is having the long list of achievement in his credit. Few of these are: during field work at site, he has completed many mega time-bound projects. Government Polytechnic Kolhapur got accredited by NBA during his tenure as a principal. He is pioneer in designing G-Scheme curriculum for MSBT Mumbai. He developed online monitoring system of Institute for MSBT Mumbai and is still in practice for deciding performance. He has organized various type of training, workshop, conferences for the teachers and students in MSBT Mumbai. He has enhanced industry relation and involved by the industry at every stage of the service. He has written book on the valuation, attended various national and international conferences. Professor Vaidya is an executive council member of ISTE since 2012, and he is presently serving as executive secretary of ISTE. He has adopted various measures to reduce expenses. He activated various programs and activities as per the policy of Honorable President and Directive of Executive Council. Professor Vaidya is working hard for development and progress of Indian Society of Technical Education. On behalf of Anjuman-I-Islam Kalsika Technical Campus, I extend my warm welcome to Professor Vaidya and take this opportunity to invite you to give valedictory address of the program. Professor Vijay Vaidya, it's over to you, sir.
Thank you, Abhisuddin Sheikh, for my brief introduction.
Welcome, sir.
Good evening, everybody.
Good.
Dr. Abdul Razak Hantogi, director of this institute, and he's well known to me also. Dr. Sharik Sayyed, coordinator. Dr. Abu Yasin Sheikh, and my dear participants. Just I was present for the last session for last some 15 minutes, and during that session, I realized the efforts taken by the coordinator and his team and management to get such a good speaker. Basically, as it said, I am from civil discipline, so I don't have much idea about the pharmacy. I only know that whenever I need some medicine, I have to go to the medical shop, and that medical shop owner or that person who is there, he will give me the medicine, and for the getting job in pharmacy medical shop or to start a shop, you need diploma. That is the basic knowledge I am having. But whatever the things they were saying, it was really excellent, and they have demonstrated the interaction and the relation between industry and academia. So if you consider our situation, this relation we don't find much at our level. Means the industry is doing their own in a different way, and academicians are doing this in a different way. For example, last some 2 3 years back, I had an opportunity to visit Singapore. So there was an international conference, and we had been to that Singapore university. So since we are from there were persons from different disciplines. They told the coordinator that we are interested in visiting the different departments in your university. So in our case, our impression was that, see, suppose if in any college or in even in our any university, if you say that we want to visit particular department, suppose if I want to visit mechanical department, if I want to visit electronics department, just we can go there and we can enter there with somebody and they will give information. But there it is not like that. Then the coordinator said that I have to take the permission from the head of the department of that section, and then we can go. And okay, we thought that that is a formality. So he took the permission. So when we went there, for entering into the door, their biometric lock was there. So unless and until you have that permission, you cannot enter into that lab. So when we entered, we were shocked because the information which they give, they told us that we are conducting various projects of industry, and they were online projects, and commercially they were doing it. Means they were taking projects on two bases. Either it is on the lump sum basis that they will give the solution and they will charge something, and in other case they will develop the solution and they will get royalty forever. So this is the way in which industry and institutes are working abroad. So I think the eminent personality which I have seen here, so and the feedback which I have just the people faculty members are given, it clearly shows that the program was really excellent. So if that program is excellent, you might have really those who are seriously attended the program, they must have learned a lot of things from this. But my sincere appeal to them is that you just don't stop here. You have to implement all those things at your institute. So there may not be 100% possibility, but at least you try your best to implement it. Then another appeal I want to make for all faculty members is that, so you have appreciated the program. It is good. It is the best program because Dr. Sharik Sheikh and others, they have taken tremendous efforts to make it successful. So now there is chance for you that you also can organize this type of program. We are having MOU with AICTE, and recently AICTE has given an announcement about the new programs for the year 21-22. So that is open on AICTE website. So you can refer that website. Even it is available on our website. So in that case, you can apply for these type of programs, and you will be getting sponsored programs in that case, provided your program is selected. The only thing is that your institute should have good reputation. Then your coordinator should take the program heading. Everything should be very good, and it should be means it should give the information about the latest practices and techniques being used in the field. Generally, it is observed that faculty members are not getting much exposure in the college because they are busy with their routine work. They are busy with their curriculum, their laboratory experiments and all. Sometimes they are getting some chance to visit industry, but that will for small duration. But here the purpose is that they should be given exposure. So unless and until you get exposure, you don't have idea what people are doing outside, what others are doing. So this is the basic concept of this, and AICT and as well as IST, we are keen on that. So whatever program being conducted, they must be conducted in an excellent manner, and the faculty should gain something. Luckily, at the beginning, when this program was sanctioned in the month of some February or March, that time it was supposed to conduct on face-to-face mode. So when the face-to-face program was there, it is very difficult to get good speakers or good resource persons, because uh, people are ready to deliver their content, but their problem is that they cannot spare much time. But luckily, because due to this COVID pandemic, it became essential to go for some other way. And during this period, we realized that the teacher community is the best community who has utilized this online facility at the best. Okay, because we were conducting our classes and all, everything was going on in different institutes. So we thought that we'll try for this online, we'll go for this online method, and AICTE also agreed, and they gave us the permission to allow institute to go on online basis. And I'm happy to say that everywhere where these types of programs are being conducted, coordinators have taken they have taken a lot of efforts to get resource persons from well-known resource persons from India as well as abroad. And ultimately, the faculty members are being benefited by this. So this is the good thing. And I think you all are maybe aware about the IST, and IST is doing a lot of activities for faculties. So I appeal all of you, those who are not life member of IST, to become a life member of IST. So you may think that what is the advantage, why we should become a life member of IST and all. So the same thing happened with me. When I joined Government Polytechnic. That time, my senior faculty in my department, there was some election, something was going on. He called me. He said, "Vaidya sir, you have to become a member of IST." But I was having total faith on him. So okay, sir, if you say, I will become the member. I didn't ask even I was not knowing what is IST, what is the long form of IST, what is does and what is the use for me. He said, "Sir, when in due course you will realize the importance of this IST, and you will feel proud that you have become a member of this organization." And now still I feel that I did the great thing that I joined this IST. So so that is why I am appealing it. And another thing is that for your information, I want to stress the point that more than 1,40,000 faculty members from different institutions are the life members of IST. We are having student chapter, under which students are enrolled in student chapter, and the students are expected to conduct different type of activities for uh, overall development for themselves. So other lot of benefits are there. And another important thing is that you have the chance to attend different seminars, webinars, national, international conferences organized by IST, as well as you have a scope to get award, because IST is giving faculty awards as well as students award in its national convention of faculty and students. So there is a good opportunity for you to go for that. So the thing is that now the time has come, because if you consider, lot of developments are taking place outside, and the technology which was there which was there today yesterday is not there today, and what technology we are using today will become obsolete tomorrow. So our students, they have to go in this field, they have to go in this situation in their field, and they have to, in the profession, and it is our duty to guide them in a proper way so that they are fit for the future world. Another important aspect is that we are not aware about the jobs that are going to come in future. So most of the many jobs are going to become obsolete, and many new jobs are going to come. New trends like artificial intelligence, machine learning, IoT, those things are coming. Robotics is coming. So everything is going to change in a drastic way, and our students should be able to cope up with all these challenges. And for that, it is very essential that teachers should develop themselves to train the two students. Now, see one thing, when you have attended this seminar, you would have learned a lot of things. So, due to which when you will start working with your students, or when you will start taking classes, or when you start interacting with students, you will remember all these things what you have heard, and definitely that will help you to guide your students in a proper way. So with this, once again, I appreciate this institute, and I know this institute, I know this campus since long, and they are very sincere and hardworking people, and they have organized this program in a very nice way. So I congratulate them, and also I appreciate all the participants for taking this program seriously. And the feedback what they have given is that the feedback is excellent, means very excellent. So once again, I appeal the institute that you can organize this type of programs in future also for the betterment of technician education, and we are there with you at IST. So whatever activities and all there will go hand in hand for the betterment of technician education. Thank you.
Thank you, Professor Vaidya, for your inspiring valedictory address and valuable guidance to us as a teacher. We were enjoying the refresher program from last 6 days, but your valedictory address made us feel that in true sense, now we have closed the loop of this program. Thank you, sir.
Thank you.
Going ahead, now I request our very own director, Dr. Abdul Razak Hanutgi, to give concluding remark of the program.
Thank you, Professor Abu Sufyan.
Welcome, sir.
Respected our beloved friend, Dr. Vijay Vaidya, sir. I'm really touched by your gesture and your time for this valedictory program, because you are the person, who planted this particular STTP to happen. For the benefit of all the participants, though Dr. Vaidya said that you all need to become life members of IST and also derive a lot of benefits out of it. And even institutional chapters, when your institution become IST chapter, then as an institution, you can derive so many benefits. It all depends on how many life members, how many members our respective institution has. So I still remember when Dr. Vaidya called, because we had submitted about 3 proposals to IST for this funding. One from engineering, one from architecture, and one from polytechnic. So then, in fact, 2 from engineering. So 4 proposals were there. And Dr. Vaidya sir was very, very kind enough to call me and tell that, sir, we have decided to give a program to your campus, because I know personally what your campus is, how your institution function. But my only restriction is that this time I can allot only 1 program. Now you tell me which is that program to be given to you, whether engineering, pharmacy, or whatever. So out of 4, he asked me to give my opinion. Then even I asked him, then he said, normally engineering, lot of such programs are being conducted, and yours is an interesting technical campus, then why not you go for pharmacy? Then whatever was there in my heart, though I am from engineering, but I said, yes, we will go for a program to be organized by School of Pharmacy of Anjuman-I-Islam's Kalsekar Technical Campus. So actually, the encouragement came from Dr. Vaidya, and not only this is the first instance, whatever the funding, be it technical festival, technical project exhibition, while he was in MSBT, so through IST, through MSBT, from various sources, he was our brand ambassador. So we are very happy to make Dr. Vijay Vaidya as a great personality associated with a historic organization called Anjuman-I-Islam. Sir, I'll tell you, today only I was talking to our president. There was a meeting. Then since that meeting was overlapping this particular program, so he could not participate. Otherwise, our president wanted to join this program to appreciate all faculty members and also your efforts. I already briefed him. Then he said that, yes, I know, because I still remember when you came as chief guest for our various programs. So I made it this point very clear for all the participants that a person like Dr. Vijay Vaidya, I found him as a real human, a human being par excellence. Not only in the field of his technical education, but ethically he is so sound that he is very open. Nothing, if there is anything to be told for the sake of development, he will put forth that point very effectively. A very righteous person we are associated with. And really, sir, you made this entire program truly meaningful by being chief guest of this valedictory program. So from the core of my heart, I thank you very much for having associated with this. So thank you very much, sir.
Thank you.
Now coming to our own, team. Yes, I am really blessed to have a team of a very interesting campus. People from engineering, from architecture, from pharmacy. The deans like Dr. Sharik, or Dean of Architecture like Professor Raj Matre, then the faculty members, coordinators like be it Abu Sufyan, or the entire faculty members of School of Pharmacy. Most of them are attending this. So they and our engineering fraternity and also architecture. So they gel with each other in such a way that I always see it in our IQAC. In IQAC, you can imagine, sir, pharmacy, architecture, engineering people sitting together, brainstorming, and always thinking of quality initiatives, how to add value to this campus, how to enhance quality in whatever we do. So this is the mix of faculty from young to experienced, and people like Dr. Sharik said, choosing this new campus to become a faculty, though he had all the opportunities to join any best industry in the country after coming from US. But Dr. Sharik, he chose to be our faculty. Now he became dean. But this shows that really our campus always attracts, positive souls. Those souls who are really pure, clean, honest, and positive. And that is the reason why, sir, good things are happening on our campus. And even during this pandemic, literally during pandemic, we have worked almost 3 times than normal times. Almost, our campus became a role model campus for entire Anjuman, because we kept on training all teachers of Anjuman. There are about 2,500 teachers of Anjuman. Our campus took the initiative to make them aware of what is this online teaching, what are the various tools and techniques, how to go about. Every kind of training was initiated by our campus, and especially I must keep on record that the initiatives taken by our faculties of School of Pharmacy towards innovative teaching, exuberant learning kind of scenario during this pandemic is really remarkable. So they're all experimenting with so many things, and that is the reason why we made Professor Abu Sufyan as the controller for Center for Innovative Teaching, Exuberant Learning. And he is really working, and his total team coordinators working wonderfully well for adding value to our campus. So as far as faculty members who witnessed this program, you all are blessed because you got introduced to resource persons drawn from reputed institutions and industry from India and across the globe. So be it Monash University or US, and all the contacts were, extremely, connected to our campus. And this is a typical scenario where at our own comfort, at the end of the day, attending to those resource person, really you all are blessed. Now, the only thing is, now this blessing, whatever you got, you must have got it from all the resource person as information, or somebody must have thought whatever you got, it is a knowledge base, or even some must have thought that whatever they shared, it is like a wisdom they are sharing with you. It all depends on how you take it. But suppose if you are taking it as information, then your responsibility is to take it to the level of knowledge and then wisdom, and then explore more through your research mind. If it is a knowledge that you are taking it as, then you give some wings to that knowledge and make it applicable and become wise. So taking knowledge to wisdom level. So faculty members, whatever you get, you are supposed to articulate by applying scientific principles, research tools, so that whatever you learn, that should not go into your data bank. It should not just go and settle into your folders. No, that should not be the purpose. Really, I'll tell you, I used to get the feedback, and on inaugural day I was there, and today the feedback which I obtained was such that you got the real-time knowledge which is prevailing in various institutions, various industries. That is how. And today, a glimpse what I had from today's last resource person, Dr. Rana Reyes. She was giving, such a wonderful, pieces of knowledge. Then that will definitely motivate you to become really great. So my appeal to all participants who attended this short-term training program is to ensure that don't keep this knowledge just as a stationary knowledge, and give some wings and let it run. And when you make it run, then it will become really applicable and relevant. Then you also develop as great minds, and you also become such great resource persons where other institutions can invite you. So everybody is capable, everybody is having all that competency, and that's the reason why you have invested this 1 week's time out of your schedule. And this invested time should not go waste, and see that you make yourself more competent and more innovative and more sought after faculty members in your field. So this, if it happens, then really the investment made by AICTE-ISTE for this program to be conducted, and the time and energy spent by our own faculty members and all the resource persons from various parts of the globe, that will become really meaningful. So it happens only when you take it to the next level of excellence. So with this, I simply quote some great thinkers. Like you must have heard a management guru by name Peter Drucker. I love to quote this his quote every time. Now he says that success in the knowledge economy comes to those who know themselves, their strengths, their values, and how they best perform. So he is giving an indicator to know themselves, to know their strengths, know their values, and how to perform in a better way. And these are the ways by which you can develop yourself to perform better. And there is another person from IBM, from IT field, Professor Lawrence. He says the only thing that gives an organization or an individual a competitive edge is what it knows, how it uses what it knows, and how fast it can know something new. So today, what happened? You know what you know, and you also know what you do not know, and then try to develop a mindset to know the new thing before it becomes obsolete. So that is all the purpose of these kind of short-term training programs. And once again, I thank Dr. Sharik Syed, Dean School of Pharmacy, Coordinator Professor Abu Sufyan Sheikh, and all faculty members, be it, Professor, I can see some names here, Imran Sheikh, Sabah Sheikh, Dr. Firoz Khan, Professor Mirza Anwar Beg, Maria Lal, and all these people, invariably, they joined hands and to make this program a grand success. So particularly at personal level, I'm very happy. And even I'm very happy to share with you that Dr. Vijay Vaidya sir already told that again you apply, sir, and he has even given some lead to bring in some agencies so that our faculty development becomes really vibrant activity. And in fact, I am yet to contact those persons, and that is all for the benefit of all our faculty members. So we keep on doing it, sir. So once again, I thank Dr. Vaidya to be here this evening, And all participants, really, you made a great, week, for our campus, and it all goes on the record. Thank you very much. And if I missed anybody to acknowledge, please, pardon me. And our management is also delighted to express their deep sense of gratitude to all of you, and all the best. Enjoy your life as a great professional. Thank you very much.
Thank you, Director Sir, for concluding remark of refresher program and also giving success mantra to follow as a teacher. I am sure our all participants will run to become a role model, most sought after academician and researcher. Now I request our Dean School of Pharmacy and convener of this 1 week AICTE-ISTE refresher program, Dr. Sharik Sayyed, to propose formal vote of thanks.
Thank you, Professor Abu Sufyan. So at the outset, I would like to welcome our dignitary, our chief guest here, Dr. Vijay Abadde, and just to reiterate what our director sir has mentioned, that he has been a very close friend of Anjuman, and he has been supporting Anjuman and the initiatives at our campus, especially for all these years. So thank you, sir, very much, and taking your valuable time in the evening and coming. It is an inspiration for all the faculty here that you've been here and supportive here essentially. So at the end of this entire a very exciting 7-day program, so I have this privilege and an honor to propose my formal vote of thanks. Essentially, a program like this cannot happen with support and what we have essentially at our campus here. Just 1 second, I'll pull up my slides. Give me 1 second. Okay, as a formal vote of thanks, essentially, so as our honorable director sir at the start of the program said that it is an honor and privilege for us to be associated with Anjuman-I-Islam, a very a pioneering organization, a very well-known organization in the field of education, doing some phenomenal work for under 147 plus years, essentially. So it is honor and privilege for me when I came back from US. As Director Sir mentioned, there were different opportunities and avenues, but the vibrancy at our campus and the mission to provide quality education to all kinds and all spectrum of society, and that was very inspiring for us, and that is what really attracted to me. So it is an honor and pleasure for me. So really from the bottom of my heart, we thank our management for providing all kinds of support and giving us an excellent infrastructure and the entire platform, especially to our honorable president, sir, Mr. Dr. Zahir Kazi, who himself is a practicing radiologist. 2 weeks back when he was at our campus, I had the opportunity to discuss our program with him, and he being a very involved person, he was very excited to hear the whole program and the schedule for the entire program. Unfortunately, today there was a general council meeting for Anjuman. He could not, or else he definitely makes it a point to come here. And along with him, Mr. Burhan Haris, who is an executive chairman for the entire board of Navi Mumbai. He has been a pillar of support and inspiration for all of us. And along with her, our secretary for Anjuman, Jia Sheikh Saab, and our very own director, Dr. Abdul Razak Munatagi, who is a source of inspiration and source of motivation for us. And he continuously emphasizes the fact that we are here to give quality education. We are here to develop ourselves. We are here to create this new generation of learners. And his passion and his dedication towards work is really infectious. For me, it is an honor and pleasure to be working under his leadership. Going forward, essentially, this event couldn't have happened with a very a dynamic team that we have. I'm very fortunate to have some talented faculty at our School of Pharmacy. Professor Abu Sufyan, you've been hearing him for the last entire week. He's the event lead coordinator managing the entire program. And Dr. Firoz Khan and Dr. Sabah Sheikh and Dr. Priyanka Sanap looking at different aspects of the program, with right from conduction of it, the technical aspects of it, and feedback, report conduction. There are a lot of work that happened behind the scene for having this a week-long program to be organized in a very an excellent manner, essentially. So a big shout out and a big thanks to all of them, and essentially the thanks goes to the entire experts who have really come in from different parts of world. All of them are very busy. I'm just looked at Dr. Rana Reis. She had a very major grant that she submitted just 2 days back. And so for all of them, they were extremely busy person, but when they heard about this program, they heard about the focus of it, they were very excited and they had come and joined. So I really thank them. Most of them had a very different time zone. So Dr. Rana Reis, for her, it's very early morning, 6 o'clock in Washington, DC. But even then, she was excited to talk to all of us. So I really thank from the bottom of my heart for all of their experts who have come in. And finally, last but not the least, essentially, the faculty participants who had come all over from India. And I'm sure we tried to put up a very good program that could benefit them. And I'm sure that has happened based on their feedback. And last but not the least, I would also emphasize the support and the funding that has come from IST and AICTE. We really thank this institution, which are being led by people like Dr. Vijay Ved. These are visionary people who have a very strong interest in supporting good institutions, supporting good quality program. So, sir, thank you once again on behalf of our campus. We'd like to once again thank AICTE and IST for the generous support. And we continue to look forward for more such supports in organizing more such faculty development programs and further collaborations. So with that, essentially, I end my form vote of thanks. Over to you, Professor Abu Sufyan.
Thank you, Dr. Sharik, for giving formal vote of thanks. But vote of thanks may not be completed unless we thank Dr. Sharik himself, our own Dean, School of Pharmacy, and convener of Refresher Program. He has pulled international talent from different countries of the globe and guided entire organizing team. So thank you, sir, for you as well. With this, we are done with our one week AICTE-ISTE refresher program. As per schedule and IST guidelines, we have released feedback and assessment link with MCQ-based question for being eligible for AICTE-ISTE certificate. Our internal certificate of the participation shall be shared with you within 2 days. However, you will get AICTE-ISTE certificate in due course of time on your registered email ID. Have a nice day. Thanks for joining this program.
Just 1 minute. Just 1 minute, sir. The certificate.
Yes, sir. Go ahead, sir.
Regarding the certificates, those who are getting qualified for certification, that is based on their attendance and performance.
Yes.
Those certificates will be issued from our side, and those certificates will be sent to you online, to you only, and you have to distribute to all participants.
Okay.
So participants will get it from you only.
Surely.
Sure, sir. Sure. So have a nice day. Thanks for joining.
Abu Sufyan.
Sir.
Yeah, I think you have been using a term called refresher course, right?
Program.
So since this was like in virtual mode, online, context. Suppose imagine if all these faculty members would have been there on our campus attending this, then they must have enjoyed, our hospitality, right?
Yes.
They missed so many things, right? So actually, if that is the case, then why don't we give a treat virtually, right? So just give me a screen sharing. Allow me, so that we will give some treat to all the participants.
You can share, sir. Yes.
Okay, because normally we do it with our management meeting also.
But real treat is
we thank Anjuman-I-Islam, thank all participants, and it's a virtual treat.
Thank you.
आंखों से भी खाया जा सकता है ना, शारिक सर?
Yes, sir. Sir, just to remind you, IST has given that option to have one online and one campus. So the second part or the second workshop will be done on campus. So definitely, this is a good treat, and this is a good invitation for them to join on campus in the summer when we all start in-campus programs.
But yeah, we definitely can assure that what you see virtually on campus will be more tastier than this.
Yes.
Yeah, it is true.
Okay.
Good.
Thank you,
everybody.
Thank you.
Rather, sir, feedback started coming. Surendra from NMIMS is saying that thank you, sir, for this wonderful treat. So they have enjoyed. They have enjoyed it even virtually.
Good.
Great.
Thank you. Are our organizing committee also there? Can we just see them on the screen here essentially for the benefit of? So we have Professor Sabah Sheikh, Dr. Feroz, And Professor Priyanka, can you open up your video essentially? So we have Dr. Vaidya sir here, Dr. Abdulrazak sir, as our own Professor, Dr. Director sir is here. Can you unmute your, open up your videos?
I think all participants, let them unmute their video.
That way we can have a good screenshot for all of us.
Yes, screenshot can be taken. Yes.
Yes. Please. Thank you so much. That will be really good for us. It will be good memories.
I'm also very happy to see Dr. Pradeep. I think, I know his principal. Dr. Pradeep Kulkarni from KLE,
KLE Hubli, yes.
Yeah, KLE Hubli.
Yes.
That was the first institute before we planned to start School of Pharmacy. So we paid visit to that KLE School of Pharmacy, Hubli, right? So your principal is known to us, and founding Vice Chancellor of KLE Technological University, Dr. Ashok Shettar. Do you know him, Dr. Pradeep?
Yes, sir. Yes. We know.
Yeah. Dr. Ashok Shettar is my guru.
Okay, sir. Okay, fine.
Okay.
Yes.
Convey my regards to your principal also.
Yes, sir. Yes. Fine. Yeah. Definitely, sir.
Yes. I request all participants to unmute your video. Still, some of you have not unmuted.
Mirza Salman Beg, unmute karo.
Okay.
Marialam. Maria Madam, जिसके नाम ले रहा अगर no, if they are not unmuting, it means they are not there, right? That is one. Maybe
yes. One of my classmate is also attending. I think he's in Delhi. Muhammad Ahmad. He's my M-Form classmate.
Okay.
So I am really very happy to see him in the participant.
Great.
Muhammad Ahmad, can I see you on the screen?
Ejaz Sayed, why he has put there is no video? Ejaz. Jayasri Patil Madam.
Okay, sir. Done, sir.
Okay.
Done.
Thank you once again, all of you. It was the presence of all the faculty members which really energized us. We look forward for more such sessions from yourself, and also you joining us for the in-campus FDP that hopefully we'll have in summer. We can see yourself personally and interact there.
Wish you all the best for your future activity.
Thank you, sir. Thank you so much, sir. Your presence really made a lot of difference.
Thank you.
Thank you to our very own director, sir, also for joining. I know he was very busy with other meetings, but he took out time to come in here. Thank you, sir, so much.
Okay.
Thank you. We'll close here, Abu Sufyan.
Okay. Thank you.